Hello everyone and welcome to a new video. So after we completed the cell wall inhibitors, the penicillin, the cephalosporins, the monobactams, the carbapenems, and the glycopeptides, now we are going to move to another family of drugs, and this is going to be about the protein synthesis inhibitors. So there is differences between the bacterial ribosomes and the mammalian ribosomes or the human ribosomes. And the protein synthesis inhibitors utilize those differences to inhibit the bacterial ribosomes without harming or inhibiting the mammalian ribosomes. And in case of the bacterial ribosomes, we have two types of subunits. So bacterial ribosomes, we have the 30S ribosomal subunit and the 50S ribosomal subunits, and they are like that. In case of the mammalian ribosomes, we have mammalian ribosomes. We have the 40S and we have the 60S ribosomal subunits, and they also look like this way. So we have here 30S and 50S in case of the bacteria. We have different types of the protein synthesis inhibitors. We have the protein synthesis inhibitors that inhibit the 30S subunit, and those include the aminoglycoside, and this is a bactericidal group, so aminoglycosides, bactericidal. And we have the tetracycline and the glycylcyclines. So glycylcyclines, and both of those are bacteriostatic. And we have the protein synthesis inhibitors that inhibit the 50S subunit, which is the other bacterial ribosomal subunit, and those include the macrolides, the lincosamides, and the chloramphenicol. And all of those are bacteriostatic. The macrolides become bactericidal in high concentrations, but other than that, those three agents or the three families of drugs are bacteriostatic. So in summary, all of those are bacteriostatic except for the aminoglycosides and the macrolides in high concentrations. In the next videos, we are going to discuss each of these groups of drugs each of them in a different video. So stay tuned for that.